All right. So the last two chapters deal with animals. Chapter 18 deals with the invertebrates, and then chapter 19 deals with vertebrates. And so what we're going to do is we're going to look at each animal phylum. We're going to learn the names of those phyla, their common and scientific names. We're going to look at different examples. We're going to look at structures that are unique to these phyla. And so this is my favorite part. So animals have certain characteristics. They're multicellular, but the simplest animals just have cells that are working sort of autonomously and not working together. Then the next group would have tissues but no organs, and then the rest of the phyla of animals deal with complex organs and organ systems. They're heterotrophic. They eat things to gain energy. They can't make their own food. They require oxygen for cellular respiration. They reproduce asexually and sexually. You don't think animals and asexual reproduction, but there are certain organisms, animals specifically, that do have asexual reproduction. Most are modal. That means they can move, as opposed to sessile. Sessile means that they're attached to something. They have embryonic development that's similar. Mitosis forms a multicellular embryo from the zygote. So, in separating out these types of animals into phyla, there are several characteristics that taxonomists use to do these separations. One is their body plan. Do they have are they vertebrates? Do they have a backbone? Or are they invertebrates? They don't have a backbone. And of course, the invertebrates are the most numerous animals on the face of the earth, and several different kinds are a lot more abundant than vertebrates. Then there's body symmetry. Body symmetry is sort of an artistic term, right? But it also refers to how the body can be divided into equal parts. All right. So if an animal can not be divided into equal parts, then we use the term asymmetry. All right. A means not, so they're not symmetrical. So there's one group, the peripheria, phylum peripheria, the sponges are asymmetrical. But the other two types are radial symmetry and bilateral symmetry. Radial symmetry is like cutting a piece of the pie. If you can cut it into equal parts through a central axis, like you're cutting a pie, then that's radial symmetry. And then bilateral symmetry is where the body can be divided into two halves, but they're mirror images of each other. And so here's the asymmetrical peripheria, the sponges, all right, that you just can't divide them into equal parts. Radial symmetry, Cnidaria and Echinodermata, you can cut them in equal parts and arranged around the central axis like a piece of the pie. And then bilateral symmetry, most animal phyla are bilaterally symmetrical, and you get mirror images of each other. Animals have sides, all right. The head end is called the anterior end or side, all right. The head. The tail end would be the posterior end, and dorsal, like dorsal fin, is back, And ventral, get in my belly, is the belly side, ventral, okay? And so those are sides. When using terminology of animals, those are sides that normally are talked about or used to describe certain characteristics. Digestive systems. Most of the complex phyla will have a complete digestive system. That means that they have a mouth and a digestive tract and then an anus that empties the waste from the body. But there is one group called the Platyhelminthes, which are the flatworms, that has an incomplete digestive system. And this is where you have a mouth and a digestive tract, but no anus. So in that drawing that I had before, you can see there, this animal has a mouth, digestive tract, and an anus, and that would be complete. But incomplete would have a mouth and a digestive tract and no anus. Incomplete digestive tract. And so the waste just is released from the body where the food came in the original. So they don't have an anus, so they have to release it back out of their mouth. Body cavities, all right? More complex animals have a complete coelom. Coelom is a body cavity, and that body cavity, if it's a true coelomate, has a lining in it that separates the digestive organs from the rest of the body. And that lining is called the peritoneum. It encloses the organs and helps hold them in place. If you go in for thoracic surgery, which is in the digestive area of your body, they cut through the skin, they cut through fat, they cut through muscle. Once they open that up, there is a skin-like covering over the digestive organs, and that would be the peritoneum. That is cut open as well. They do the surgery. They would automatically go back and sew that peritoneum back up before they would sew everything else up. It's very important to keep an eye on patients that have had thoracic surgery, because if that peritoneum gets infected, it becomes a dire health situation, and you could die from peritonitis, all right, which is the infection of the peritoneum. So they usually keep thoracic surgery patients for multiple days just to make sure that there's no peritonitis. And so we've got true coelomates. These are animals that have a true coelom, right? They have a peritoneum. Pseudocoelomates have a body cavity, but they don't have that peritoneum. So the digestive tract and the reproductive organs, etc., are not separated. And then there is they're acoelomate animals that when you cut them in half, they just have muscle tissue. They don't have a cavity at all. It's all filled with muscle and organ. So in our picture here, a true coelomate would have this peritoneum that separates the digestive tract from the rest of the organs. Acoelomate, you can see I've drawn here, just has muscle tissue all in it if you cut it in half. And then pseudocoelomates, which are represented by one phylum, that's Nematoda, have opening but no peritoneum, right? So all of the organs are together. And another characteristic that we can separate phyla from each other is segmentation. This is where body the body is divided into segments. They may look the same or they may not look the same. If we go back to our sides picture that I drew right here of an ant, the head, thorax, and abdomen are body segments. They don't look the same, they don't have the same functions, but they are segmentation. We think, most of the time when we think of segmentation, we think of like earthworms, right? Redworms. But other organisms do have segmentation as well. So what we're going to do now is we're going to go through each of the phyla, right? And we'll have the scientific name, the common name of the phylum, and then we're going to look at characteristics that are only found in that particular phylum, and also structures that are found only in that particular phylum, and then maybe some of the examples of those phylum. And so the first one, the group is Porifera, and they're the sponges. They are the most simple of the animal phyla. They do not have symmetry. They are asymmetrical. So this is definitely unique for sponges. They're only phylum that is asymmetrical. We'll see an actual sponge here in a second, and you'll see how you just can't divide it into equal parts. They don't have tissues or organs, so they have a mixture of different types of cells that are working on their own. They're not working together for a function. They're functioning separately. Since they don't have organs or organ systems, they don't have a digestive tract. They are acoelomate. They have no segmentation. But they have these specialized cells called choanocytes. Sometimes they're called collared cells, like a collar on a shirt, right? And we'll see that in just a second. And these choanocytes have flagella, and these flagella allow for the water to form a current. And as the current is moving through the sponge, then particles of food are deposited in the sponge, and cells absorb that food. They are sessile. That means they are attached. They don't move. They're not motile. And they're filter feeders. They filter water to obtain their food. And so they have a specialized support system. They have these things called spicules. Spicules are made of either calcium or silicon. They're spines. They give the animal support. There's also protein fiber called spongin that allows for support as well for the sponges. And there are calcium sponges. Those sponges have just calcium spicules. They don't have spongin. Silicon spicules, likewise, just have silicon spicules. But the ones that you think of when you think of sponges are bath sponges. And bath sponges have spicules, either calcium or silicon, and they have spongin fiber as well. These are what you think of when you think of commercial sponges. And they are asexual and sexual in reproduction, and most are marine. There are a few that are freshwater, but most of these are marine forms. And so a choanocyte looks sort of like this. It has a cell body and a collar, and then has a flagella. And the colony of a sponge will look like this. It has an opening, and then they have these little openings on the side, those pores, and those pores lead into canals. And these canals are lined with either choanocytes all right, or amoeboid type cells. There are also the spicules, those spines, and then those protein fibers called spongin.